All right, so let's talk about this self-excitability business. How do cells of the SA node end up having action potentials periodically without a stimulus? Okay, well, in red are your SA node fibers, your sinoatrial nodal fibers. And what you can see is, I like to start on the downstroke, okay? So we've had an action potential depolarization, now we're repolarizing. So the membrane potential drops below the threshold. Now what's interesting is this slope right here, okay? What's happening is when this membrane potential drops, but then all by itself it starts going back up again. Why? Because of leaks, okay? Sodium and calcium leak in during this upstroke. That causes the membrane potential to rise all by itself, okay, because you've got positive ions that are coming in. Now, when the cell reaches its threshold, which is usually about minus 40, okay, when it reaches its threshold, that is then would typically cause sodium channels to open, right? And the sodium channels would cause the depolarization. But because we're sort of up here in the minus 40 region instead of down here in the minus 80 region, instead of sodium channels playing a role, it's actually calcium channels that are playing a role instead. L-type calcium channels close. First they open, right? They open at threshold, and then they close about 100 to 150 milliseconds after opening. That's this point, right? Because when they close, then repolarization is going to happen. You see? So, no plateau, right? We just learned yesterday about the action potential and how it's supposed to look like this. Well, in conducting system cells, they don't contract. They don't need the plateau. They don't need the calcium to come in and trigger contraction, right? So, yes, it's still calcium, but it's not playing the same role. It's just purely ionic. Okay, so then the cell repolarizes, same thing happens. Leak of sodium, and then fires again. Well, one of the things that we'll see is the sympathetic and parasympathetic nervous system, the way it controls the heart rate is by controlling how deeply these cells repolarize, and therefore how long it takes for the leak to result in another depolarization. All right, so the autonomic nervous system, it affects this baseline and therefore affects heart rate. So now we know why SA node fires all by itself. Leaky sodium channels, right? Okay, so from there, from the SA node, we go into the internodal pathways. These fibers are found in the walls of the atria, okay, and they evolved to conduct signal, not to contract. So the pathways, the internodal pathways, are very rapid. So signal can travel very quickly through them. As the signal travels past, the area around the internodal pathways is then depolarized and activated. All right, so it's a little bit like watering a lawn with one of those hoses that has lots of little holes in it, right? The water goes through the hose really fast, but the water leaks out along the way, and that's the depolarization signal. It's leaking out along the way as we're going through. So there's three of those internodal pathways, an anterior, a middle, and a posterior. They all arrive, boom, 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 internodal pathways, they all arrive at the same place at about the same time, and that's at the AV node. The AV node has many fewer gap junctions than other parts of the conducting system do, which means that the signal for contraction, the depolarization, the membrane potential change, is conducted through the AV node much slower than it is in other places. That's on purpose, right? Evolution gave us a delay system. So the signal arrives here, but then it takes a long time to pass through the AV node, 100 milliseconds, which really isn't very long, but it's long compared to how fast it's taking the rest of the cells. All right, then once it's through the AV node, then it picks up speed again. It actually becomes even faster than it was in the atria. So the impulse is 0.16, 100 milliseconds is how I remember it. And it's to give a chance for the atria to actually contract before the ventricles contract, right? So you can see the difference. We go from 0.03 delay to 0.12, right? So that's a big difference. That's 0.9. That's almost 100 milliseconds. And then from there, the whole rest of the system is only 0.04 more, right? So the rest of it is very, very fast. See, 0.12 and then 0.16. That means that only 40 milliseconds happens between exit of the AV node and passing through the bundle branches. So that's very, very fast. So we go SA node to internodal pathways to AV node and then into the ventricles. All right, so the ventricles have much more muscle mass than the atria do. Okay, you can see that anatomically. Because of that, there are a lot more cells, and each cell has more mass. So it needs rapid conduction in order for it to meet our characteristic of all the ventricular cells contract at once. Okay, so we have high speed conduction. The AV node splits from the into the left and right bundle branches. These are large diameter, non-contractile fibers, very fast conduction. In other words, they have lots and lots of gap junctions. So it runs through like a wire, 150 times faster than the atria. So very, very fast. The action potentials in this system, similar to what we see in action potential in the nervous system, they can only travel in one direction, through the AV node and down. The reason for that is it's very similar to nerves, where there's a refractory period. All right, when the contractile cell has repolarized and is in a hyperpolarized state, it can't send another signal. So it makes it so there's only a one direction that's possible. I say at least normally so. Okay, one of the things with heart, this is called electrophysiology, what we're talking about, right? And one of the difficulties is when this goes wrong, it can go wrong in a whole bunch of different ways. Okay, so you can have pathology states where action potentials are traveling the wrong way. Okay, even from ventricle all the way back up into atrium. Now, those aren't compatible with life because usually you're ticking along at 25 beats per minute, right? But that can happen. It just isn't the normal. All right, bundle branches terminate in Purkinje fibers. Total time between AV node and the end of the Purkinje fibers, 0.03 seconds, 30 milliseconds. So very, very fast. And then, just like our hose with all the holes in it, that very fast water flow then triggers depolarization of the ventricular cells all around it, and we get contraction then. So our spread of conduction is well timed. Remember, I said in my first slide, we need to have the atria depolarized and then the ventricles depolarized. We need both sets to depolarize together. You can actually see that in the numbers. Okay, so first at time 0, right, SA node and interatrial pathways, very little in time, so 0.04, 0.3, 0.06, those are all very small numbers. So the atria are all depolarized before we get to the ventricle, which starts at 0.16. You see that? So what I'm showing you in this picture is that, in fact, the atria contract first, and they contract at about the same time, and then the ventricles contract, and they contract at about the same time. Because see, all the way at the end, 0.19, 0.19, by 200 milliseconds in, the whole heart is depolarized, and we're ready to contract. But you can also see the difference. So the atria are ready at 0.07, the ventricle are ready at 0.19, which means you have like 120 millisecond delay for the atria to contract before the ventricles contract. See, it fits into what we were just talking about. So it takes time for the signal to move from SA node on down. Longest delay is right here. See, we go from 0.03 to upwards of 0.16 after that.